Okay, welcome. Excuse me. Okay. Welcome back to protein synthesis. Now we're picking up in part two. All right, part one, we had big picture, all of like the whole thing of protein synthesis. Now we're going to go into a little bit, okay, so maybe a little more detail. All right, so let's go back to the making of messenger RNA and the modification of messenger RNA. So that's what we're going to talk about today. So this making of messenger RNA, you want to remember that this is occurring in the nucleolus. So this is happening in the nucleolus, and the nucleolus is like a copy machine where we're making a, sorry, I don't know what's going on here, making a cheap recyclable copy of the DNA sequence. All right, so there's three phases of production to a transcription unit. That is a piece of messenger RNA. So here's transcription, where through this process. So we're making DNA into the messenger RNA code. That's what's happening. So here's our DNA, and gene 1, gene 2, gene 3 in here. Now we're taking out one piece and taking a look. Okay, so if we took out one piece and took a look at what does it look like? So here's the DNA and 3 prime, here's your 5 prime N, here are your nitrogenous bases here, and then we're going to transcribe that into messenger RNA. Now notice we don't have, instead of T's now, we have U's. And so we're doing the, and then 3 sections right here is the codon, or it's also called a triplet. All right, so the 3 phases of production of making the transcription unit. Whoa, let's go back here. The first one is called initiation. And this is where we actually build the factory to make the messenger RNA. So basically that's what we're doing. So we're going to see the factory of enzymes at the bottom here. We're going to see all kinds of factories that are, so we're building this. So a protein called a transcription factor, so here's the transcription factor, attaches to the promoter sequence. So here's a promoter that's being transcribed. Okay, so the transcription factor attaches to, so you can see here, here's our promoter, and our transcription factor is attaching to it. Then additional transcription factors, which are proteins and enzymes, are added in the building process. So additional transcription factors are going to be added in this process. Now this whole factory is called a transcription initiation complex. So you can see down here, transcription initiation complex. So transcription, you can actually look, if you listen to, look, I mean, just think about the term. Transcription initiation, so the start of the starting of transcription, and it's the complex that's going to happen. So transcription is the process that's being done, initiation is the beginning process, and complex indicates that we have many parts involved in the making of this structure. And you can see that there's a lot going on here. All right, step 2 is elongation. And if you think about, if something is elongated, it means it's getting larger, and so it's stretching. So this elongation process is referring to the actual making of the messenger RNA. We're adding nucleotides in here. So this must be done, must be done in the 5 prime to the 3 prime direction. It has to be done in that direction. So you can see the direction of the transcription. You have RNA polymerase 2 that separates the DNA double helix to make room to work and adds the nucleosides to the growing molecule. So these nucleotides here are added in. So you're going to have RNA polymerase that's separating it and then attaching these nucleotides. So cells can make these multiple copies of RNA because the DNA is left intact. This DNA stays the same. You can see here's our bubble. So it's opened it up. This is happening right here. And this is protected within the nucleus. And then this messenger RNA is going to leave. And then the third step is termination. Well, just like it sounds, it stops the transcription. It terminates the process. So a stop codon is made for the ribosome. Remember when we looked at the chart last time, the chart showed stop codons. So there's a start codon and there's a stop codon. And so the stop codon is made for the ribosome, and the factory molecule actually slows down. So you have your RNA polymerase. The RNA polymerase slows down until it stops transcription by forming an AAU-AAA sequence, and then is released from DNA. So that's the sequence, the AAU-AAA. This is the termination signal. So once it reaches that, then it's released from the DNA. Don't worry about memorizing this sequence. It's not going to be on the test. What you want to do is understand what's happening in each of the steps. So you have initiation, elongation, and termination. So think big picture, what's happening in each one of these steps. And the purposes of telling you about the termination signal is just so that you understand what's going on, but don't get bogged down in the details and the memorization of the steps. All right. Now we've transcribed. So this is post, it's after transcription and the modification that's going to take place. So the changes that are going to take place. So we have this primary transcript. So our original transcript of messenger RNA is going to be modified, and we're going to end up with a secondary transcript. All right, so here at the front end, the 5' end, there is a protective cap is added here. And this would be kind of like you putting on a hard hat to protect your head when you go outside in a construction site. So there's a protective cap here, and there's a tail. All right, and then on the end, the back end, the 3' end, a poly A tail is added. Poly means many, and A is for adenine. So there's 50 to 250 adenines that are going to be added onto the tail here. And if you think about it, the more adenines, the longer the tail, right? Well, the longer that messenger RNA is going to last. So what this does, the caps and the tails end up as protection against the digestive enzymes that are in the cytoplasm. Because remember, the messenger RNA is now going to leave the nucleus and it's going to go out to the ribosome. And in order to go to the ribosome, there's digestive enzymes. Those lysosomes are all in there to digest, and digestive enzymes to break things down in there. And so you want protection for your messenger RNA. And then, so you have this, so that's the front end. So here's your front end, and then there's back end protection. Now in the middle here, so in this middle part, right in here, you have these exons and these introns. And notice what's the difference here. You can see that the introns have been excised. They've been cut out, and these exons have been spliced together. So we consider, what we currently understand at this point is that introns are non-coding, that we like to say that they're maybe not as important as the exons, because this is where the coding region is in the exons. All right, so during this step, we're going to remove, during this middle modification, we're going to remove those non-coding introns that act kind of as spacers. And we're going to use these things called spliceosomes. A spliceosome, think about the name. If you're splicing something, you're cutting something out. So it's a type of enzyme. It acts as a scissor. So, and then we're going to rearrange the separated coding exons. And the rearranging allows for different sequences of exons. So it allows for a different order to take place. And a good way to think about this is to use the exons A, B, and C, and you could make the following sequence from it: ABC, ACB, BAC, BCA, CAB, CBA. So this rearranging increases the number of proteins or enzymes that can be made, but without increasing the amount of DNA. So it increases the options without having to use more DNA. It makes it far more efficient. So what the spliceosomes do is they stitch, we can consider it stitching together the pieces to make a finalized piece of secondary messenger RNA. It's now going to be ready for transport to the ribosomes. Okay, so we've gone through transcription here, and now here we're going through modification of that. Here's our protective cap and tail, and then those introns are spliced out of there. We basically stitch the exons together, and now we're going to go out through the nuclear pore out into the cytoplasm. Notice we have our cap and our tail here to protect as it goes out into the cytoplasm out to the ribosome. That's all we have for today. Oh, just hang on. I promise you we're going to get more detail on this next time. We're going to finish up the story. Have a great day.